Thank you very much, and thank you everybody for getting up and making it out. So as I understand, this is an educational session, and we were just talking up here about how interesting the actual topic of the session is, and that there's not a lot of data yet to describe things, but there's enough data around to suggest that it's actually an exciting time that's coming up. So I will try to sort of just casually walk through some ideas, some thoughts, and some learning objectives, and I will leave plenty of time for questions, and we can just sort of chat at the end. So I thought I would lay out a few learning objectives that if you get nothing else at this early time in the morning, at least get these few things. First of all, that the dominant sequencing technologies that are available for human genomic studies and the structural information they can capture. Most of what my lab does is look at the structure of the genome and how that impacts the function of the genome, and a lot of the added value for these long-read sequencing technologies is really to capture structure that's missed by short reads. The relative value of emerging long-read technologies from the perspective of the genome. Where in the genome are these technologies actually benefiting us? I think it's a really important question. We get very excited by new technologies, new ideas, new flashing lights. But what we often try to do is benchmark things and understand exactly what are we capturing and what are we missing from each technology. And then the selection process, really, to consider when thinking about whole genome sequencing approaches to human disease. So as you're laying out your studies, or the field is thinking about the way forward and the amount of sort of our total investment portfolio that we are willing to put into different technologies, how should we be thinking about that? Okay, whole genome sequencing and structural variation. I will focus almost entirely on structural variation over the next 40 minutes or so. And so let's just start with that baseline knowledge. I'm sure almost everybody's familiar with all of this, but the idea being that these are genomic rearrangements that encompass large segments of the genome, greater than 50 base pairs. And quite often they have been described in terms of copy number variation, so deletion, where you delete a segment, or duplication, where you take a segment and duplicate it, either in tandem or in a dispersed manner. We then have multi-allelic CNVs, or multi-copy state CNVs, where we can have the same segment repeated many times. However, from the analyses that we've done so far, I'll say that this only represents maybe 80 to 85% of all structural variation in the genome. And in fact, there are many other classes of structural variation that are quite important. They're not as common as CNV, but they can rearrange the genome in very interesting ways. And that includes insertions and inversions and translocations and complex rearrangements. And I think for a very long time in the field of human genetics and genomics, copy number variation, or that was attainable through karyotype or microarray, was really the dominant type of variation that we could look at. And now we have new technologies to look much deeper. So over time, technologies have really driven our new insights, and particularly when it comes to the structure of the genome. So from microscopy, of course, we had 50 years or so of cytogenetics, where we could look through a microscope and look at the karyotype, look at the chromosomes. In the early 2000s, folks including Mike Wigler and Jonathan Sabot and Charles Lee, and Steve Shearer and Charles Lee and others developed microarray technology to really look at large gains and losses of DNA throughout the genome. This led to really amazing insights, I think, into human disease architecture, particularly the effect of duplicating or deleting large swaths of the genome. And there were clear signatures of disease association when that was done. And then really we moved into the sequencing era almost 10 years ago now, where the sequencing allowed us to capture the information from these submicroscopic CNVs, as well as the very large chromosomal rearrangements, but also get to nucleotide resolution, or in the early days, near nucleotide resolution, with Jan Corbell and Mike Snyder's original sequencing of large fragments, and then moving into sort of the 1,000 genomes era of deep sequencing. So what's next? As we gather data and we start to think about how we're going to interrogate the genome moving forward, the question becomes, what's next? When we have these biobank scale and population scale genomics data sets that we're putting together, how should we be interrogating those? How should we be looking at the data, and what do we hope to extract from all the genomes that are coming? I will focus entirely on 3 technologies. So I'm just going to touch on them here. Mark Chasin yesterday, I know, gave a nice session on new technologies and how to deal with them. And so I will not cover sort of the methods or the technologies in too much detail, but really focus more on the genome and where the reads go and what they look like. But so we have short read sequencing. This is basically dominated by Illumina sequencing, where you get this clonal amplification of a given cluster. These are small fragments. You can then read the ends of the fragments, and the entire idea is no matter what the size of your fragment, you can sequence the ends and put millions and millions of ends together to reconstruct the genome. Pacific Biosciences is they have a technology, single molecule real-time sequencing, and the idea there, it's been out for actually quite some time, but what's happened recently is it's become more tractable in terms of cost to actually be used in human genomic studies at some sort of scale. And the idea is basically that in real time, you're actually sequencing a DNA strand as the DNPs are sort of added and a nuclease or a polymerase reads off. You can sort of, every nucleotide that gets added, you can read it with a camera as it's moving in real time and extract that information. So as you can imagine, this allows for single molecule information to be captured. But of course, the error rate is a good bit higher than if we're clonally expanding a given fragment set. And then nanopore technology, which I think a lot of people have been interested in the last couple of years. And the idea is moving DNA through a nanochannel where you can sort of read off the sequencing based on the currents of an individual nucleotide. Okay, so I'll walk through each of these and just give a few points on their interest and then sort of get into some of the meat of what we can get from them. So short read sequencing, it really dominates the current landscape, and I think there should be no sort of bones about that. While the newer technologies are very exciting, almost all the data in the world from whole genome sequencing right now comes from short read sequencing. So it's used in virtually all large-scale whole genome sequencing or WGS studies, and is really being adopted in clinical genomics testing, or at least is being benchmarked early for clinical diagnostic testing. The benefits are low cost. The costs have been dropping precipitously over a decade, and they're now really in the range of where kind of microarrays were 10 or 15 years ago. They are standardized methods. You almost get the same genome from any of the major sequencing centers around the world, very low error rates and extremely high capacity. Negatives are the short reads. So we have a huge genome, over 3 billion bases, and yet we're trying to piece it together with 300 base pairs at a time, which is really 150 base pairs from two ends of a small fragment. This presents a lot of challenges. It is not an ideal way to analyze or to assemble a genome. It's really limited then in reference-free applications. So here are some of the basic metrics. You get 300 base pairs. It's around $750 per genome, about 0.1% error rate. There's the largest published human study. I thought it would be important to sort of benchmark where we are. I think maybe there's a misperception of where some of these technologies are and how much work has been done on them. So the TopMed study is on bioRxiv right now. It's 53,000 genomes. There's about 140,000 sort of sitting around the CCDG TopMed consortium. And then, of course, there's many, many, many hundreds of thousands and millions of genomes that are out there that have been funded or already completed. I estimated 2 million here, but it's actually probably closer to 4 million if you consider sort of what's being funded right now. So there's a lot of genomes. Okay, the long read technology. So let's start with the Pacific Biosciences. I'll refer to it as PacBio throughout. There's two modes of this. A newer chemistry allows for this consensus circular sequencing, but the technology up until now has been this idea of continuous long reads, or having that fragment and sequencing around it to get as much sequencing data as you can from as long as the fragment is. The CCS sequencing now is actually really improving the base quality. So the concept there is to sequence around a fragment multiple times so that you get every nucleotide a number of different times, and then you can sort of do some error correction. They're starting to look sort of close to Illumina reads in terms of base quality. So that's actually really exciting. But for right now, the benefits, virtually all data is on the continuous long read, so that's what I'll talk about today. These are long reads. They are less biased to given error modes that, say, Illumina sequencing is, and there is improving base quality, but they do still have very high error modes. The costs have been astronomical up until very recently. They require high input, and it's a complex technology, so it's not like what I described with Illumina, where every lab will produce the exact same results. Some basic metrics, the fragment lengths and reads are maybe 5 to 15 kb, sitting right around 10 kb, and for CCS, this is pretty tight, around 10 or 11 kb. We're at about $11,000 for a 30x genome. At least this is on the Broad sequencing platform when everything's incorporated. Somewhere around 5,000 or 6,000 for a 10x genome. And the error rates are quite high, like I said, 13 to 15% for subreads in the old TLR mode, although they're getting to less than 1% for CCS. So that's starting to get actually very encouraging. And the largest published study, so PacBio has been around a very long time, and we talk about this a lot. There are a few studies out there, and this may not be actually the largest right now, but Evan Eichler, Pete Adano published a paper in Cell a couple of months ago on 15 genomes. And so if I estimate sort of the number out there or available, it's somewhere around 100 to maybe 500 if we consider all the reference consortia and the other cohorts that are being sequenced and sort of put out there that the community can access. So it's very different from millions, but we're not at a couple of genomes like we were a couple of years ago. Okay, and then the other is nanopore technology, as I talked about. We have this benefit of getting very long reads. Now, they're not all very long. In fact, most are very short, but there are some extremely long reads, and these are really exciting as we get to sort of megabase length reads, what we can do with those. And so, getting through repeats, no bias in GC, and other significant benefits, still very high error modes. So the kind of per base nucleotide error rate is quite high. So you're not going to be able to trust your individual nucleotide coding variant, SNV, but to get very long reads and be able to reconstruct the genome is actually very exciting. And the read lengths are extremely inconsistent, and a lot of this has to do with how hard it is to get the right fragment lengths. And so the costs are dropping. So we're estimating around $3,000 right now, although we don't have this as a product at the Broad Institute, so I don't have a tightly benchmarked cost. The error rates are still high, and at this point, I think the largest study is 11 samples. And out there in the community, certainly less than 100 that you can get to and download. Okay, so here's a look at the technologies. And this is just a quick plot from Karen Garamella in the group, looking at sort of the PacBio CLR, CCS, and nanopore data from NA12878. And sort of if we look at the 10 KB mark, which is very exciting. Remember, with Illumina data, we're talking about 300 base pairs. So at 10 KB, somewhere around 90% of the data or so is better than 10 KB in terms of length. And then while the number of fragments, the percent of fragments that you read actually drops significantly when we move beyond that, as you can see, particularly for Oxford nanopore data, I mean, we do start to get very long reads. And there are a number of individuals that are out there sort of saying, all right, well, let me try to capture this long part of the genome, and so everything that I sequence is actually very long reads, and there's molecular methods to do that. So this is exciting. The complement of that, of course, is this plot, which is the idea of capacity. And so as we think about kind of the estimated yields from a given machine in terms of gigabases, you can see that the capacity is almost entirely dominated by Illumina, and particularly with the NovaSeq S4s. And so this is a slide from Mark Chasen from yesterday that I'm just showing again here, where this is sort of the state of single molecule long read sequencing. In 2015, Mark and others had the haploid cell lines. 2016, we added a few more. 2017, we added a few more trios with our Human Genome Structural Variation Consortium, which I'll talk about a good bit, the HGSV. And then 2018, a few more. And in 2019, what's coming, we know there are studies out there. Not huge numbers, but in the 10s to 20s, maybe even up to 100 genomes, I think will possibly be published this year. And so this is where we're moving. So again, exciting technology, exciting topic. The current literature is not actually replete with a number of amazing new insights from this technology. So I think that's why this is actually a very timely session in sort of thinking about what's going to come over the next couple of years. Okay, so this is just a summary of that, sort of from the educational standpoint, just to have these backgrounds, the read lengths, the fragment lengths, and sort of the number of genomes out there from millions to hundreds, or maybe 1,000 to tens to maybe hundreds. Okay, so now what I actually really want to talk about, which is the value of these technologies and what we can think about when we talk about what we're going to capture from them. And I think the properties of the genome really must underlie this discussion. So I just want to make sure we're all on the same page. And I know a lot of the genomics folks out there will know this, but I don't know that this is necessarily common knowledge for people who don't often sort of align reads to genomes, is that when we think about the genome, about half of it or so is really unique sequence that appears only once in the reference that we have. And so when I'm putting these numbers up, by the way, I'm talking about the human genome reference that we have, not necessarily a given assembled genome. About another 40% is repeat mass sequence, but a sort of broad category of repeat mass if you go to UCSC and turn on the repeat masker. So they're sequences that are seen elsewhere in the genome, and they're repeat masked under various headings, but they don't qualify in sort of segmental duplications or simple repeats or long tandem repeats or these sort of highly repetitive sequences. So everything I'm going to talk about today is really going to combine these two together, if you will. And so that represents about 90% of the genome. So about 90% of the genome is either unique or mostly unique. And then about 10% is a smattering of different types of really highly complex regions. Segmental duplication, simple repeats, like I said, long tandem repeats. These regions that you think about as sort of really repetitive heterochromatic regions and things like this. So the relative value of these technologies is really dependent upon your scientific goals. And we have to keep that in mind as we move along, because there's a lot of talk of long reads in the clinic and interpretation. But if we think about what I just said, in fact, and then talk about where the genes are in this space, well, you know, of this unique and repeat mass sequence, almost all of the genes also live in that region. And, you know, somewhere around 96% of all constrained genes, so genes that we know to be intolerant to mutation and underlie most of Mendelian discovery, are all sitting in this sort of unique and repeat mass regions, as well as almost all OMIM genes. So somewhere around 95% of this unique sequence harbors most of the genes that we're going to interpret in any sort of meaningful clinical way. Now, that's not everything. Certainly the segmental duplications and simple repeats, and I'm going to use SD, SR a lot, so just so you get used to that. SD is segmental duplication, SR is simple repeat. They're depleted for genes, and particularly constrained genes. The one sort of caveat I'll make to that is these are really what we know under the lamppost, right? They're the only places we've been looking. They're the only things we've been able to access, so it's very possible that there are, in fact, more evolutionarily constrained or intolerant regions in the genome that we've never looked at. But at this point, what we know and what we interpret clinically or in basic research or disease association are sort of within that 95% of the genome. So again, what is your question? That is really the relative value of the technologies, and I'm going to touch on this over and over again. So I am going to talk about 3 data sets largely, and so I'll just orient you to them now, and then I'll revise them later. The first is GNOMAD, or the Genome Aggregation Database, and a structural variation data set that we did in that database from 15,000 genomes. The other is the Simons Foundation from Autism Research, and so this is an autism cohort of quartets. Very useful to parents, a proband and an unaffected sibling, so that you can sort of benchmark what happens in a disease state and also what happens in an unaffected individual from the same parents. And these are all Illumina sequencing, and then this HGSV, the Human Genome Structural Variation Consortium, which is 3 trios and basically every technology available. And so this is just a look at that. I won't go through this in any great detail. Again, Mark talked about this yesterday, but this is the idea of 3 diverse trios. And really every sequencing technology that we could use, we did. And so we sequenced these sort of to the depth that we can and analyzed them as deeply as we possibly could from a large consortium of individuals, each with expertise in various areas. But really here we have PacBio, Illumina short insert. We generated Illumina large insert, 10x Chromium, BioNanogenomics, TrueSeq, HiC, and then we actually at the end generated some Oxford Nanopore data. Okay, so I'll come back to this cohort, but this is just to give you an idea of sort of the data that we have and the various fragment lengths and total coverage. So we're talking about somewhere around 580x coverage of 3 genomes or 9 genomes, 3 probands. Okay, so now let's talk about the value of these technologies. And I'm going to start with short read genome sequencing. As I said, this is really the largest data set out there, and tell you what we've already extracted from the genome, which actually, you know, it may not be entirely clear that in 2019, there actually wasn't a lot of good references out there for structural variation. So if we think about the gold standard reference for single nucleotide variants and indels, I think pretty much everybody knows the Exome Aggregation Consortium, GNOMAD, dbSNP, these resources, and what they've catalyzed, right? Over the last couple of years, as we've moved to 50, 60, 70, 100, 200,000 exomes, we've really completely improved our understanding of human demographic history, of mutational constraint. What regions of the genome are evolutionarily constrained against mutation and why? Where is the refined clinical interpretation within these regions? And really, we have now these databases for these frequency filters for rare diseases. We even have a sort of a database of human knockout identification. If I ask you what the resource is for structural variation, can anybody tell me this? And turns out that really the most complete study up until now is the 1,000 Genomes Still, which was published, Ryan Mills and others, in 2011, and then an update of it in 2015 from Sudman et al. And this is really 2,500 genomes, 4 to 7x coverage, very low coverage, almost entirely focused on copy number variants. And from sort of technologies and algorithms that are, a half a generation old or so now, at least 5 years old in terms of what was done, and yet that's our best resource. And so unlike the SNVs and indels as well, the 1,000 genomes data is available, but you can't really browse it and extract the information that you may want to compare to a 30x genome. Okay, so that was really the goal of GNOMAD-SV. So there was a genome aggregation database from Daniel MacArthur and others that EXAC was the first incarnation, and then GNOMAD, which includes genomes. And so our goal was to take all of the GNOMAD data and try to fit structural variation algorithms to it and release the data openly for the entire population to go and be able to use it as an open resource. And so that's what we did. I will not focus at all on this other than to say structural variation detection from Illumina short read sequencing is still a huge pain in the behind. We had, in the HGSV, we have a huge group. Basically every algorithm that we used was run by the original developer, and yet the concordance among algorithms, I took it out just because I didn't want to get into the detail, but the concordance amongst algorithms was actually terrible. And it was somewhere when we took the concordant algorithms and looked for the influence of different types of data that we were able to start to begin to capture something that we believed in. So in GNOMAD, what we've used is these algorithms here. So 5 of them deal with paired end and split read information. So do the reads map to different places, or do they read right through a breakpoint? 3 of them deal with read depth. So if I don't have a breakpoint, say 2 segmental duplications came together and there's no way for me to tell where in the genome they are, of course, if I lose or gain a segment, I can count the reads in that region and sort of normalize that depth. So we combine all of these together into a single pipeline that's now on FireCloud, and WDLs are written, and so it can be sort of routinely implemented. Okay, the data set is 14,890 samples from many, many different studies that we've aggregated all together. The first thing we were scared of is could we actually joint cull all of these? And it turns out we could fairly well, so we only lost about 700 genomes. And so the QC worked out fairly well, and we end up with about 14,200 genomes, which is about a 6-fold increase over the resources we had. This PCA plot is based on structural variants, not SNVs and indels. So from structural variation alone, we can sort of easily separate populations and demographics history. Not surprising. Okay, what does GNOMAD have? So the total data set is actually about 500,000 structural variants. And I think if nothing else, the one thing to take even from Illumina sequencing, and the one thing to walk away from here, is that the vast majority of all structural variation that we can see in the population is small and rare. So this is 10 kb and above, and this is on a log scale. So you can see almost all of the variation is less than 10 kb, and in fact, almost all the variation is less than 1 kb. The median call was about 387 base pairs, and we're still not very good at extremely small variants, so in fact, it's probably even lower than that. Almost all variants are rare, so somewhere around 40% of the variants in GNOMAD are 40 to 60% are singleton within a given individual, or rare less than 1%. So most structural variation is rare, and most structural variation is small. So what we were doing with microarrays or technologies to get fairly large events was really missing most of the variation in the genome. And I'll get into PacBio long-read sequencing and where there's huge benefit there. Okay, so copy number variants, all these other variants that we thought we could get from other types of sequencing is fine. What about complex structural variation? So my lab is actually, one of the things I've been very interested in for a very long time is complex rearrangements of the genome, going back to sort of when I first started as a postdoc and we were doing sort of long insert sequencing, and in cancer they were finding these extraordinarily complex shattering of the genome and what was dubbed chromothripsis at the time. We were seeing very similar patterns in the germline of autism patients that ended up being very, very similar, in fact, to chromothripsis, except they were occurring in sort of a balanced manner without large gains and losses of DNA. And Wieger Kloosterman and Edwin Koopman in the Netherlands were seeing very similar patterns. And the point I want to make here is that these variants are attainable from short read sequencing. In fact, at the time we were doing long insert sequencing. And over time, we have sort of further refined them, but they're accessible. So beyond copy number variants, we can get these types of highly complex rearrangements, and we see them in the general population. So over the years, this was work from up to 10 years ago now, or at least 8 years ago, of these incredibly highly complex rearrangements that we were seeing in various studies and documenting. And so this information is there from short read sequencing. And one of the things that we can do from GNOMAD is ask the question, how often does it actually occur in the population as well? And so I put this slide up here because I grabbed it from a talk that I did at American Society or ACMG or one of those meetings back in 2015, where I said, hey, we're finding all these inversion-mediated things at very large scale, you know, megabase size and hundreds of KB, but we have no background reference set in which to interpret them, and so that's what we need. And here we are in 2019, and we actually still don't really have a great background reference set. So that was one of the major driving motivations of GNOMAD. And so now with GNOMAD, we have some set of background. So we basically have 6,000 fully resolved complex structural variants across 13 mutational subclasses. So ranging from this kind of inversion with paired duplications to deletion, inversion, duplication, various CNV-mediated events and inversion-mediated events. And the one thing I want to point out here is they're fairly large. So all genomes have these complex rearrangements. They tend to be fairly large in the order of tens of KB or more, and they're very strongly selected against. So singleton, proportion singleton is our measure of how strongly selected against or evolutionarily constrained a given variant is. So the more selected against, the less often it'll rise to higher frequencies in the population. And each of these, as you can see, has a very high proportion of singletons. Okay. So our pipelines capture about 8,200 high-quality structural variants per individual from Illumina sequencing, and you can see that here. So this is a population, and every line is the number of variants that we get in a given individual. So you can see it's extremely consistent across individuals. And of course, it follows human demographic history, just like you would expect. Africans have more structural variants than Europeans. This is about where we think we are with Illumina short-read sequencing. So just to keep this in mind as we move forward. And one of the questions that we had for this and our autism work is, as we generate these structural variant maps, are they sufficiently sensitive and sufficiently specific? And I think in the context of this session, we really need to frame that question in terms of short-read versus long-read. So for short-read sequencing, we compared to chromosomal microarray in 2,000 individuals, and we find 99.7% sensitivity. In our autism work, we actually took every de novo prediction. So there were 171 predicted de novo events from 519 families, and we did this sort of cross-site validation where we tried to design an assay for every single variant that we predicted to be de novo, and our molecular assays validated 97%. So it looks like, from the perspective of sort of disease association studies, we can now routinely capture structural variants with higher specificity, actually, than we can indels. So this was much higher than our indel validation rate, which was about 84% cross-site blind. So this is, you know, encouraging that we've moved to the point where structural variants can be part of our panel of variants that we consider in every study. Compared to the state of the field right now, so sort of over the years, we've had a number of studies on structural variation. This is GNOMAD. This is our Simon Simplex collection work, so this was that autism work. Here is the 1,000 Genomes project at about 4,400 variants per genome. CCDG, which is a compendium of studies, is generating about 4,000 structural variants per genome. So you can see that somewhere between 4,000 and 8,000 structural variants are sort of routinely captured in every single study at this point in the most recent couple of years. And this is where the short-read sequencing, I think, leaves us. Okay, I'm just going to zoom through this. Let's just focus on, of the loss of function variants in the genome, we have about 200 from structural variation that we weren't seeing before or weren't capturing when we were just looking at SNVs and indels. And so if you just look at this plot right here of predicted loss of function variants, this is what the SNVs and indels are, and this is what the structural variants are per genome. And then if we consider very rare structural variants or very rare loss of function mutations, it turns out that about 25% of all rare and private loss of function mutations per genome in GNOMAD are actually represented by structural variants. So we get about 18 SNVs and indels and about 6 rare or private structural variants per individual. So in fact, when we're thinking about disease association and clinical applications, without capturing that background structural variation load, we're missing about 25% of the known predicted loss of function mutations. And then just as an idea of singleton proportion, here's all autosomal structural variants. And as I said, this is a measure of selection. And you can see basically every class of structural variant that disrupts any part of a gene is actually very strongly selected against compared to the background. And so that's just the point to be made here. And of course, we get very, very weak when we talk about regions around genes that aren't coding. Okay, and I'll skip this just to say that constraint from structural variation correlates very highly with constraint from predicted loss of function mutations from SNVs and indels. And one of the really interesting points here is this is copy gain. So this plot is SNV and indel constraint, and this is matched against copy gain and duplication. And what you can see is a very high correlation across all genes. And what this means to us is that the genes that are dosage sensitive to deletion by measure of constraint or intolerance are actually very dosage sensitive to duplication as well. And this background sensitizing factor actually occurs throughout the genome at every tranche of evolutionary constraint. So duplication is an area that we're certainly focusing on because it hasn't been as well described as deletion, but it's actually quite important. Okay, and then I'm going to skip this other than say the variants are in the browser. Go to GNOMAD, download them. They're completely open use. You can download the structural variants. You can look at your given gene or use the data for any type of study you want to do. It's completely open access, and there's no publication restrictions at all. The paper's on bioRxiv, but we haven't published it yet, and you are free to publish on whatever you like from the data. Okay. So back to the long reads. So that's where we are in the area of short reads. So now I'm going to spend the rest of the time talking about long reads and the comparison between these two. Okay, so back to this data set that I talked about, the Human Genome Structural Variation Study and all the technologies that we threw at it and how we analyzed it. Again, Mark Chasen talked about this yesterday. The only point to this slide is to say when we have Illumina sequencing, we use a reference, we align the data to a reference, and then we call structural variants that look different from the reference. With the long read sequencing, we're allowed to go through a series of, and it's complicated, but a series of complex phases that really allows us to assemble the genome. So a given individual, you can put their genome together in a haplotype and phase resolved manner so that the structural variants that you capture, you actually know their phase. And from the Illumina genomes, with the kind of most sensitive output we can get, we were generating about 10,000 structural variants per genome, or 11. PacBio, we were getting 24 to 25,000. So even at that background rate that I was talking about of 8 to 10,000 structural variants per genome that we weren't capturing, there's at least double that again that we can capture from PacBio sequencing. And this is the really exciting part. So we have these 24,000 structural variants per genome. And the question is, and this is what I want to spend the rest of this sort of session on, where are they? Where are these coming from and why are we missing them? Because if we're talking about clinical genomics or we're talking about sort of million sample short read sequencing, I think that if that's the data that we're going to have right now, we really would like to know what the long read sequencing is telling us that we're missing in that short read sequencing. And that's really been the goal here. So this is just that look. I mean, as you can see, the long read sequencing completely dominates the structural variation detection from short read sequencing. I mean, this is a major improvement. So where are these copy number variants and structural variants coming from? And so that's what Jufeng in my lab has been working on for a while now as part of the HGSV, is asking where do they come? Okay, first of all, overlap. I think this is really one of the most important points, is when we look at just raw overlap of deletions and insertions, and I'm going to treat deletions and insertions very differently. So for PacBio, we're calling sort of duplications insertions, whether they occur in tandem or are dispersed, and it's a whole nother discussion. But really, if we look at the shared space between these, we see that, as you can see, there's a lot more PacBio than Illumina and a lot less shared than you would like to see. And this is particularly true for insertions. Again, let's go back to the genome and what I talked about at the beginning. Remember, about 90% of the genome is unique and repeat masked. And I'm going to talk about these two together as sort of unique plus RM. And then we have all these other sort of repeat-mediated type of variants up here. Remember, this SD plus SR segment is 0.1% of the genome, and SR alone is 0.6%. So simple repeats alone are 0.6% of the genome. When I look at the Illumina data, what I see is, in fact, this 90% gets reduced to about 45% of all deletion calls in Illumina in this space, whereas about 50% or so of all Illumina calls are actually in this tiny space up here, so the repeat mask sequence. When I look at the PacBio calls, only 28% of them are actually in this 90%. And in fact, the vast majority of the PacBio calls are in this tiny region up here. So if we blow this out to where the actual structural variants are occurring, they're occurring in these repetitive sequences. And this data is from haplotype resolved structural variation detection from Mark and the HGSV group. So I'm sure there's many improvements that can be made on it, but it's kind of the best that we have right now in the field. And Mark has done a wonderful job with this, with Tobias Marshall and others. Okay, so we have all these changes in the genome from PacBio sequencing, and we really wanted to ask, can we figure out what the error modes are, or can we at least look at the features of the raw data and figure out what's unique about them, or can we calibrate what's detected in PacBio and not detected in Illumina. Are all these repeat-mediated structural variants just, are they just artifacts? Are they false positives? Are they things that we're never going to capture anywhere else? And so one of the ways that we thought about this, I'm showing you one plot here of depth. We've actually done this in a number of ways, including with recurrent paired end changes or split reads. So imagine you have a read that goes right across the breakpoint. You can ask the question of how often do I see that versus see normal reads and get some distribution there. But what I want to explain here is depth. And so this is the median coverage for a deletion from short read sequencing. And this is the flanking region. So imagine you have a deletion, here's the breakpoint, and I really want to know what the depth is at that breakpoint compared to the rest of the genome, and I'd really like to normalize that by the local coverage because the local sequence context will define a lot. For deletions, we see about half, right? So this actually looks very good. So the deleted region, when the short read and long read sequencing is concordant with each other, about half the coverage as in the flanking region. So this looks really good. Where we see the same variant in long read and short read sequencing, our depth profiles look quite nice. When there's unique variants in short read sequencing, certainly a fraction of them look real. We get a nice little bump at 0.5, but then you see it gets much dirtier. So there's no real change in depth within the region flanking the deletion or duplication, the deletion as there is at the breakpoint. And then when we have variants unique to long read sequencing, there's almost no depth profile at all. So by doing this in a number of ways, this is just depth. We did the same thing with paired end. We did the same thing with split reads. And then we went back to the raw data with a program that Yufeng developed while she was in Ryan Mills's lab called Vapor to actually go to the raw PacBio data and go look for signatures of breakpoints and then sort of do a recalibration of the variants after that. Okay, so this I think is the take-home message from all of this work, which is, again, if you remember that our genome, this was 90%, we're now down to about half. But this is the Illumina plus PacBio deletion. So this is really where those variants were concordant. Still, we get about 30% of the variants are actually ending up in repeat masked simple repeats, and about 17% in seg-dup plus simple repeats. But as we move towards PacBio unique variants, you can see that they basically all live in this repetitive sequence. So as the Illumina data is becoming more unique, it's always hovering around this kind of half of the variants existing in unique and repeat mass regions. But the regions that Illumina is failing to capture are very much sitting in these repeat-mediated sequences. So these are the PacBio deletions, and then these are the deletions that are only seen in PacBio data. And you can see only about 9% of them are actually in unique or simple repeat mass, and the rest are in these highly repetitive regions of the genome that the short reads just are not accessing. I mean, I think that's the take-home, is they're just not getting there. So as we think about sort of overlap, and this is the last point, and the idea here is the orange is variants unique to Illumina data, the blue is variants unique to PacBio data, and then the green is shared. And so I showed you that plot to begin with where very little was shared, less than half of all variants. Well, it turns out as we start to break the genome apart and move down into these regions, which as I said is the unique and repeat mass houses 95% of the protein coding genes and all the OMIM genes and all of these things, we're getting actually very high concordance there. With our pipelines, with the Illumina pipelines. And once we do the recalibration of the PacBio data and the Illumina data based on those profiles I talked about, we actually end up at 98.6% concordance in the unique and repeat mass regions only. As soon as we move to these repetitive regions, it's a mess. This is deletions. This is the other point. So if you're asking a question about a unique region, a genic region, and a coding region, not a ton of difference in structural variation detection between the two. If you're asking a question about insertions, it's actually very different. So when we look at insertions, far more insertions or duplications are called in the unique and repeat mass regions from Illumina than were the deletions, and the concordance is actually very poor. So when we look at concordance between Illumina and PacBio, even in the unique regions, we actually only see about 64%. So about 30% or a third of the PacBio insertions in completely unique regions of the genome are still blind to our Illumina algorithms. Whether it's the data or the algorithms, we don't know, and we're working on that, but we're still missing them entirely. So even if I gave you a map and said, sort of, here be dragons in the genome, worry about your Illumina data, but these places look very clean, in fact, that turns out to not be true for insertions. About a third of the time, Even when I have an Illumina read in a unique region, I'm missing an insertion that I really should be capturing, and that's the most optimistic after recalibration. If we just take the repeat mask on its own, it's only about half of the insertions. So there's a huge miss here in terms of the insertions in the genome. Okay, so I will end there in general and just give you a couple of take-home messages and have 5 minutes left to make sure we leave plenty of time for Anna. So structural variants in the human genome are really enriched in these repetitive sequences. They are on a background level enriched in the repetitive sequences, whether we're talking about short reads, long reads, whatever we're looking at, these regions of the genome are highly enriched for structural variation. The structural variants that are unique to long-read sequencing really largely reside within multiple classes of repetitive sequence, the STRs and the segmental duplications. And this is especially true for deletions, where the phased, assembled structural variant deletions that we find from the HGSV are almost entirely captured in the Illumina data as well. This is not true for insertions, where, again, the short reads fail to capture about 1/3 of the insertions discovered by long reads, even in unique and sort of fairly unique repeat mass sequences. So if we want to ask questions about the added value of long-read whole genome sequencing, really it's that the value here as I see it is that we can get variants that we've never seen before. The fact that I said before, we think that most of the disease architecture actually lives in these unique and repeat mass regions. Well, you know, on some level we haven't ever shined a lamppost on the other regions of the genome. We don't know what parts are intolerant. I think the long reads will really open up a lot of work into repeat expansions, insertions, deletions, and repetitive sequences. You know, if we think about sort of the triplet repeat expansions and the trinucleotide repeats and the polyglutamine disorders, there are certainly many other repeats that we're missing. And I think maybe against the backdrop of the fact that Chelsea Lowther, actually today, I meant to mention this earlier, I thought I did, is going to talk about our work on whole genome sequencing and prenatal diagnostics in the 1 o'clock session this afternoon. And if you're interested there, it sort of tells you from the short read sequencing what we can capture in the prenatal space. And in fact, it turns out from sort of the most severe developmental disorders to sort of run-of-the-mill autism of unknown etiology, we're only solving somewhere between 15 to 40% of the cases, and including even the most severe cases. So there are certainly, I mean, a lot of that, of course, is polygenic and complex, but there's certainly other factors there that can be captured, and the long reads is certainly one approach to do so. But I think we need to be honest about this. You know, there's a lot of talk of the hope of long read to solve cases, and I am encouraged by that, but there are not very many long read genomes out there, and there's very little example of sort of new discoveries of disease pathogenesis from a long read genome that wasn't captured from a short read genome. There are anecdotes. There's certainly an example here or there, but when we talk about really hundreds of thousands of patients and how to sort of best diagnose them, there is not a lot of evidence of long read sort of getting to sort of pathogenic variants. We have a few, and one of them is in X-linked dystonia Parkinsonism that we published a while ago, but there's not so many examples. So I will leave it there. And just thank my structural variation team at the Broad and in my lab at Mass General, as well as our many, many, many collaborators. And almost everything I talked about was a direct collaboration with other labs. And really, it's been a lot of fun. Thank you.